In this video, we'll explore what live blood analysis is and examine the impact the atom cell device had during this type of analysis. Live blood analysis is a screening method that allows us to observe the various components of a fresh blood sample under a microscope. It provides insight into the functionality of the blood system and can reflect aspects of a person's overall health. Human blood is made up of approximately 45% cellular components, namely red blood cells, white blood cells, and platelets, and 55% plasma. Blood performs many vital functions, including transporting oxygen, regulating body temperature, and supporting the immune system. The condition of the blood often correlates directly with our overall health and can be influenced by diet, lifestyle, and environmental factors. During a live blood analysis, a small drop of blood is obtained using a finger prick. The sample is placed directly onto a glass slide and viewed immediately under a microscope at up to 1,000 times magnification. The lighting technique used allows even the smallest details of the blood to be seen clearly. This method enables practitioners to observe the structure and behaviour of the blood components in a near-living state, similar to how they function inside the body. The atom cell is designed to be worn on the body to help protect the wearer from the potential negative effects of background electromagnetic radiation. To assess its effectiveness, we conducted a study using live blood analysis, focusing on exposure to a Wi-Fi router. The aim was twofold. First, to determine where the radiation from these devices affected the blood, and second, to evaluate whether wearing the atom cell had any measurable impact on the results. In the study, an initial blood sample was taken from the test subject. The subject's food and fluid intake had been carefully monitored, and it was confirmed that they had not been exposed to a Wi-Fi router prior to testing. The first sample revealed a healthy blood profile. Red blood cells were freely flowing, with intact cell membranes and clear plasma. 30 minutes later, the subject sat in front of an active Wi-Fi router for 20 minutes. 10 minutes after exposure, a second blood sample was taken. This sample showed a compromised blood profile, with red blood cells clumping together, noticeable rouleau formation, the stacking of red blood cells, and restricted blood flow. After another 30-minute interval, the subject was again exposed to the active Wi-Fi router for 20 minutes, this time wearing the atom cell on the body. 10 minutes later, a third blood sample was taken. The third sample showed a marked improvement. In fact, the blood profile was superior to the initial baseline. Red blood cells were no longer clumped, rouleau formation was absent, and normal blood flow had been restored. These results suggest that the atom cell may help counteract the negative effects of Wi-Fi router radiation on blood health. The full dataset from this study is available for viewing on the research page at atomlab.uk.